Okay, well, thank you very much for having me. Let me tell you a little bit what we're doing at Gero with respect to applying physics of complex and dynamic systems to aging and very large human data sets in order to understand the nature and character of human aging and understand the limits of age reversal. So as you may know, aging in different species is very different. We have in humans a situation where mortality increases exponentially and doubles every 2 years. And this does not need to be like that in nature. We have examples of other mammals, such as naked mole rats, where on the bottom right, you can see that the mortality is a function of age, is pretty much constant. This is the results from Calico study. So essentially, what we need to understand is what is the biology, what are the medical, lifestyle, and any other factors that are controlling this diverging behavior. So at Gero, we use very large data sets of animals and human data. Quite a few years ago, we started from tens of thousands of mice, medical histories, published a bunch of papers, got partnered with UK Biobank, where we have access to now 500,000 electronic medical records of fully characterized individuals from the UK. We have guys with proteomics, genomics, and everything else, published another bunch of papers, established an industry collaboration this year with Pfizer. We are now having access to 10 million electronic medical records from the US, and the total amount of medical data that is available for this kind of studies is now approaching 100 million. So in a way, we are living in the best of times where humans finally became more studied animals than any other species on our planet. We have more human data than everything else. So we have a very simple idea in mind, is that since whenever you see an exponential acceleration of anything, we can think of that something as an example of exponential instability. So essentially, we are studying different organisms with a simple idea. Whether those organisms are stable dynamically, does it mean that errors get amplified exponentially, produce more errors, and these animals disintegrate exponentially and die? So these guys would be kind of simple age, standard agers, like normal agers. Or we were thinking that the regulatory networks of some animals may be more stable in such a way that their regulatory errors have no time. They eliminated too fast. They don't have any time to produce more errors. And then in those cases, we should expect animals not to age. And the basic question was if this idea is true and where are humans on that scale? So as I said, we have access to lots of longitudinal data, and that's very important because the dynamic stability cannot be studied in cross-sectional data sets. So we have longitudinal data and we train our algorithms in order to fit the longitudinal aging trajectories of different animals to solutions of differential equations that describe aging. And in mice, by the way, things did turn up very simple. On the central graph on the bottom, you see the network output. You see the variable that you can extract from the data. And in an unsupervised way, that variable is simply the variable characterizes the health state of a mouse and predicts the future state of the mouse. And that variable is actually a frailty index, so it's going up with age. It's going up with age exponentially. It correlates to all hallmarks of aging, and the doubling time of the total amount of damage that you can measure in the mouse is actually the same as the mortality rate doubling rate, which means that aging in mice is indeed a simple exponential instability. The red dots on this curve are the frailty indices of animals of all ages that are too sick to stay alive in the lab. They're killed for ethical consideration, and you can see that mice literally die in labs because of limiting, or by reaching the limiting biological age. By the way, these frailty indices, if you compute them in this way, they correlate to hallmarks of aging like senescent cell burden and everything else. So, well, mice are literally dynamically unstable, which means that if you introduce any regulatory error in a mouse, you will end up having more regulatory errors in the same animals that would accelerate exponentially. That sounds scary, but at the same time, it means that there is no homeostatic equilibrium in mice, which means that if you find a drug that would reduce hallmarks of aging in an animal in a short time, the animals would still age at the same rate after the application of this intervention, but they would never catch up with the control, which means that in unstable animals, you can do systemic rejuvenation. And that's, by the way, exactly what we see. So, for example, in our experiments, we see that if you treat animals with experimental anti-aging interventions, so this is time-resolved biology in a way. So on the left, you have the damage senescence, which means the frailty index. This is the computer-generated measure from blood analysis in these animals. And on the horizontal axis, you see the age in weeks. And we have had 2 interventions against aging, just 2 shots of experimental intervention between the weeks 72 and 82. And as you can see, since there is no any drug in the circulation, the treated group is forever below the control, which means that if, as I said, you have an example of unstable, dynamically unstable animal, you can always produce rejuvenation. And that's why we believe that in the coming years, you will have almost infinite number of papers where any intervention acting against almost any hallmark of aging in mice will produce lasting effect. This is another example. So this is the thing that we are actually trying to push into preclinical studies. So this is another experiment. Now this is the survival experiment. You can see on the horizontal axis, you have the age of the animals. Time equal to 0, the scale is weeks, days. The time 0 here is 100 weeks old. So these are geriatric mice. So on the vertical axis on the left, you have the survival. And you can see that these animals are dying from natural causes. So the control is dying. And once again, we have had an example of a single shot intervention, time-resolved biology again. You can see that there is a mortality delay that is comparable with the mortality delay produced by rapamycin that we know works well in aged mice anyway. Interestingly enough, that these animals do not only live longer, these guys are also healthier. We have lots of hallmarks of aging measured in these animals. We have anything functional, frailty index, we have rotor-rot experiments, open field experiments, and everything else. But if we're looking at the effect of the larger size, this is the reduction of senescent-like cells in the immune system of this animal. So in a nutshell, aging mice is very simple. They're literally dynamically unstable. They exponentially disintegrate. And since they are unstable, it's very easy to produce rejuvenation. And we have examples of rejuvenation. Other people have examples of rejuvenation. Everyone who is working with mice and aging has examples of rejuvenation. Now, you can use the same methodology in humans. And here we were up to a great surprise. So actually, once again, we acquired lots of data from consumer lab diagnostic companies, so blood analysis from different countries. We generated, we obtained the data, including multiple measurements from the same individual. And then we started looking at the trajectories of the biological age in these animals, or any number that could be associated with biological age in humans. I will show you the actual data in a moment, but the surprise was that in humans, there is no single variable, no single physiological variable that is going up exponentially as we age. As I said on the first slide, aging in humans is best defined as exponential acceleration of mortality. So definitely we have exponentially increasing chances to die. But once again, there is no single physiological measure that is going up exponentially. In mice, as I told you, almost anything that you can measure is going up exponentially, and the exponent is exactly the same as the mortality rate doubling time. So humans are not the same. And this, by the way, big problem because you have the exponential phenotype that is mortality, but you don't have exponential physiological markers, which means that you don't know why humans have exponential mortality and what you should be doing in order to reduce the exponential mortality. So this is a schematic representation. I will give you more graphs in a moment, but basically anything that looks like a biological age in humans, instead of going up exponentially, it's fluctuating around the mean that is going up a little bit, and the range of the fluctuations is going up a little bit. And then at some point, very late in life, after 50, 60, 70 years, depending on stochastic, you have a situation where the markers of biological aging in humans become apparently increasing and running away. So essentially, humans are built to be robust. We're not mice. We control our hallmarks of aging for the most of our life. And then late in life, there is a situation where the hallmarks of aging get out of control and we become more like mice. So humans actually hold a very peculiar position between negligibly senescent animals and normally aging animals. We are almost like naked mole rats for the most of our life. And then very late in life, we start disintegrating. So how does it work? Once again, you cannot drive such a conclusion with cross-sectional data. The cross-sectional data gives you smooth curves, and this doesn't work like this in humans. I think some of us know that from our own experience. As we grow older, our equilibrium weight, for example, does not going up smoothly and increasing. You have a situation where your equilibrium weight is something, and then within a year, you have a change to another level of something. And this is not reversible, by the way. So it doesn't matter how much diet or exercises you do, you would not return to the previous value of your weight. So humans are not aging along a continuous aging trajectories. You have a bunch of shifts, and those shifts are accumulating in such a way that you have a continuous trajectory in cross-sectional data sets. So what we have done is that instead of measuring, doing regressions to produce biological age or anything else, we were looking at how physiological variables in humans change along the individual trajectories. And interestingly enough, we found, as I said, that biological age is fluctuating. The fluctuations are fast, which means that human organisms are stable most of our life, and the recovery time, which you can measure from the autocorrelation function of the fluctuations, the recovery time is actually increasing. So as you know, when you are young, you can party on Friday and pretend to be working on Monday, but as you get older, you cannot do that anymore, which means that your resilience, your ability to recover is actually decreasing. So we measure the resilience directly. So on the right graph, the resilience is the recovery rate. We measured the recovery rate from longitudinal trajectories of the biological age in humans as a function of age. We did it from two sources in order to compute the biological age. We used blood markers, blood cell counts, and we also used signals from wearable devices like this. As you can see, it doesn't matter from which source of the data you are measuring the recovery rate. The recovery rate in healthy humans is of order of few weeks. The recovery time. And as you can see, the recovery rate is going down as we age, linearly, by the way, which means that at some critical age, at about 120 years old, the recovery would be impossible, which means that the loss of recovery, the progressive loss of resilience, is actually the phenotype that is limiting the maximum human lifespan. So on the left, you can see the metaphoric representation of these findings. Essentially, you can think that your physiological variables fluctuate in such a way that they fluctuate in around some kind of minimum in regulatory free energy surface. And the shape of this regulatory potential is changing as you age. So the barrier, the rift that is protecting you from disintegration is disappearing linearly as we age. The range of the fluctuations is increasing. You spend less and less time in your best functional state. So that's why it sucks to be old, but fluctuations can throw you away out of the equilibrium, and then chronic diseases set in. So once again, to understand how it works, this is the metaphoric representation. Once again, the horizontal axis is your whatever favorite marker of hallmark of aging in humans. Most of your life, so let's start it again. Most of your life you are stable, which means that whatever happens with you, you get back to the equilibrium position. But as you age, your ability to recover is destroyed, and then stochastically at what point in your life you get unstable and disintegrate. And this is, by the way, not just a metaphoric representation. So from wearable data, you can actually see this process happening. On the right, you can see the fraction of people who are not resilient. So we measure the recovery rate in humans, and we find those guys who have recovery rate more than 3 months. These are pretty sick guys, by the way. And you can see that at any age, the number of such people is going up exponentially with age, and the coefficient, the exponent, is exactly the 8 years. So it coincides precisely with the mortality rate doubling time. So this is the exponential phenotype in humans. Stability is the late life phenotype that defines the frail. It doesn't matter what you do. So you can take methylation in proteomics, in transcriptome. You do on humans, you always you look at classic image. Most of the time, it explains most of the variance in any kind of human cross-sectional data. And all these kind of parabolic clusters have less they are following their future. The shape is actually the consequence of non-linear coupling. Linear with age, so this is the damage. So all the linear features in human signals are the damage signals. And the parabolic shapes are the cluster of features that are responders. So those are the systems that respond to the stress that is produced by the damage. So the stress responses are reversible. You can revert them in the same way as you do it in mice experiments, and the linear features are not reversible because they are not a single process, they are entropic. And by the way, the linear process exactly measures the entropy. So it's a thermodynamically irreversible process, which means that since it dominates the data, most of aging in humans cannot be technically reversed, although it can be stopped. That brings me essentially to this kind of slide, is that in humans, contrary to mice, in humans, there is no single aging phenotype. In humans, there is one entropic component, the damage component, that we, for the lack of a better word, is now calling the true aging. And then there is, of course, a frailty. In humans, we can be aged, but not frail. In humans, we can be not aged, but you can be frail. These are two different things. In mice, this is the same, or technically frailty sets in so quickly that you cannot see true aging in mice. That means, by the way, that mice are very bad models for aging in humans. So frailty occurs late in life in humans. It can be reversed. So of course, we can help people who are very sick and very old already. And of course, by controlling that feature, you can do maybe up to 10 years life extension in humans, but not more, because in humans, hallmarks of aging and frailty are controlled for almost all our life. They're only important for the last maybe 5 to 10 years, which means that if you act against those features by drugs that work in mice, most probably you will end up with things that work only in very sick people and produce very small life extension. And this is, by the way, what we see already in clinical trials. So most of the longevity industry and academic groups are actually working on this phenotype. Then there is another phenotype, which is true aging. This is whole life phenotype. It's irreversible, but it can be stopped. And if controlled, it could produce multifold extension of human health span and lifespan. And this, of course, focus our work, if we're not talking about preclinical development of actual drug candidates. So this is just a simple summary of that. As I said, there are two aging phenotypes that work in a very different way. So, for example, if you have a drug that works well in mice, most probably it's a drug against frailty in humans. It will have transient effects. By the way, you know that if you have fasting or smoking, these things do change lifespan in humans. But if you stop fasting or if you stop smoking, your chances to die actually get back to the norm. So which means that if you do a little bit of fasting and then stop, it doesn't have long-term effects on your lifespan. So you have to apply such interventions lifelong. And even if you do that, the effects on human longevity will be small. If you act against damage, if you act against the entropic component, that actually controls the resilience. If you control resilience, you have a chance to slow down aging, and the effects of such interventions would not disappear. So the longer you apply them, the larger would be the effect. So with that, we have been predicting that for already a few years, and we keep saying that the drugs against any hallmark of aging will reduce essentially all other hallmarks of aging in mice, and you can even produce rejuvenation in mice. The very same drugs would produce very small effects in humans, and the effects would be transient, meaning that larger effects would occur late in life. And that's, once again, we enjoyed recently a conversation with Brian Kennedy, who is doing clinical trials of alpha-ketoglutarate, and the results there are practically the same as we suspect. And drugs that slow down aging in humans will have small effects in mice. By the way, mice are bad models, but those drugs need to be developed because this is the only way to produce strong interventions. So how do we actually do that? As I said, we are working with longitudinal data. So the largest data sets of longitudinal data are the data sets with electronic medical records, because there you can easily have tens of millions of people. You have CPRD in the UK, you have lots of commercial providers in the United States. It's not very difficult to obtain that data. And we're training very large models on those data. So we're training predictive models. They are unsupervised, which means that we don't know what we're going to get there. And these models actually compress the state of human health in a certain point of time and use those variables to predict the future state of human health. And those things, if done correctly, output two different measures for you. So one of them is the biological age, which is literally the total amount of damage. I mean, the biological age on the left graph simply correlates to the total number of diseases that you have diagnosed, as it should, of course. The correlation is not perfect, so you cannot say that medical records explain all the biological aging. Yes, by the way, you cannot say that methylation explains all of the biological age. This is not a regression to biological age, by the way. You cannot obtain such a measure with a regression. And on top of that, you can also diagnose a state of a number of functional systems that interact with each other by means of coupling with the biological age. So on the right graph, you have the percentage of these systems broken. This is an unsupervised model, so it predicts that there is a certain number of functional units in a health body, in a human body, that can get broken. And here on those graphs, you measure the number of pathological states as a function of age. You can decode those functional units back into the diseases because you operate in electronic medical records. And here you can see that there is a cluster of metabolic diseases that is going up in population as we age, and the doubling time of that cluster, this is an exponential cluster, which has the largest variance, and the doubling time there is exactly 8 years old. 8 years, which means that the metabolic health is the weakest functional unit in the human body, as we have been suspecting. And this is not senescence, by the way. So as I told you that in mice, the phenotypes that are associated with inflammation and senescence killing the animals. In humans, as we know, I think every doctor knows that the exponential phenotype in humans is the cardiovascular, is associated with cardiovascular diseases. So once we have these phenotypes that are obtained by large neural networks, we can do standard genetic analysis of these things. And on the right, you can see an example. So you have 3 genome-wide association studies. The top 2 are the genome-wide association studies of our phenotypes from our neural networks. The top one is the biological age, and the middle is the metabolic health cluster. And then on the bottom, you have meta-analysis. I mean, this is published data, not from us, by Peter Joshi and others. This is the meta-analysis. This is the meta-analysis of parental longevity GWASs. And you can see that our models actually have genetic loci either in the cardiometabolic cluster or in the biological age that match almost all loci that were discovered by investigation of parental longevity. The important thing here is that now we know which of those loci are describing aging and which of those loci are describing disease-specific mortality. As you can see, most of the parental longevity loci are actually the genetic variants associated with cardiometabolic health, as it should be, because at the age of our parents, most of the people were dying from cardiovascular events. But of course, cardiovascular mortality cannot explain all mortality. As I said, mortality is the effect of longevity and diseases. So these networks and this methodology that we're developing is actually helping us to differentiate the effects of aging and disease. As you can see, the genome-wide association study of the biological age is not empty, which means that we have genetic variants to investigate with the hope to control human aging. So that's basically it. If we at Gero and with our academic friends are performing investigations of human longevity with the idea to control human aging, we do pretty much hardcore aging research. We are trying to count the aging phenotypes in humans, compare those with animals. We do target discovery using very large neural networks and very large data sets of human data. This brought attention of pharma because pharma doesn't want to do drugs against aging, but pharma wants to do drugs against chronic diseases, and they want to learn how to control for aging, and that's why they're calling us as experts in the field. And of course, we, as a side product of all these developments, we also developed the biomarkers of aging and frailty. in order to first define phenotypes and use them for target discovery, but I hope eventually also for clinical trials. So I would complete my presentation here. So if you have any questions, I would be delighted to answer if I have. I think I have no time. I'm sorry for that.